Hello, and welcome to GATK. My name is Derek, and if you came here looking for a place to learn about the DRAGEN-GATK pipeline, then you've come to the right place. We have got a fantastic program prepared for you today, and we hope to give you what you need to get started using DRAGEN-GATK, along with resources that you can reference long after this webinar is through. In this webinar, you will learn what DRAGEN-GATK is, you will learn how to use it, and you will have experience using it for yourself. Coming up, we will have presentations, demos, and opportunities for you to ask questions to honest-to-goodness GATK experts. However, for those of you lacking the general fortitude to engage with 2 hours of continuous online content, I will present to you our take-home message. DRAGEN-GATK gives you the ability to analyze your genomic data in essentially the same way that big biotech company hardware does. However, it's free and open source and available at gatk.broadinstitute.org. Now, let's get on with an overview of today's workshop. First, I'm going to wrap up this introductory section of the webinar by giving you a crash course on the tools and a taste of what's to come. Next, James Emery is going to present on the DRAGEN-GATK pipeline for variant discovery that we'll be using. And I should mention that James is literally the expert on this topic because he wrote the dang thing. Moving on, Michael Gatzen will explain to us how we can determine that the DRAGEN-GATK running on my 5-year-old laptop is able to produce the same output as Illumina running code on their proprietary FPGA hardware. Again, Michael's definitely the expert when it comes to DRAGEN-GATK and functional equivalence, so we're all very lucky to have him present for us. Next, you're going to actually get your hands on the DRAGEN-GATK pipeline for yourself, and you're going to be able to use it, you're going to be able to take it home with you and make your own outputs. Finally, if there's enough time left over, and I think that there should be, we'll have an opportunity to answer questions from you, the audience. We'll be joined by Geraldine Van der Auwera. She literally wrote the book on GATK, so we are expecting lots of good questions from you. So I guess what I'm trying to say is we have a star-studded cast here as far as GATK goes, so if you have any questions, go ahead and post them in the chat and we will address them in the Q&A section. Also, keep an eye on the chat for helpful links that we're going to be posting there as well. For example, links to the workspace or additional resources that you can use later on. And if you're watching this on YouTube, all of the relevant links can be found in the description box. Don't worry if you think of a question after the webinar is closed. That's fine. All you have to do is visit us on our forum, go to gatk.broadinstitute.org and click on the forum link. What we have done is actually created a special DRAGEN-GATK webinar discussion board just for this event. We'll be keeping an eye on this board moving forward, so if you have any issues running the pipeline, this would be an excellent place to post. And while you're there, don't be a stranger. While the GATK forums are a resource, they're also a community, so we definitely encourage collaboration. For example, if you engage with other users and help troubleshoot their issues, we can even promote you to the rank of GATK Guru, earning you a fancy badge so you can show off to all of your friends. It's more than just a fancy badge, though. It's basically a mark of approval from us on the GATK team, showing that you've become a GATK expert in your own right. And so it's definitely something to be proud of, and we look forward to seeing you there. So why don't we just go ahead and start at the beginning? What is DRAGEN-GATK? It all starts with DRAGEN. DRAGEN is a genomic analysis platform produced by Illumina that uses specialized hardware-accelerated processors to run genomic analyses quickly and accurately, even in difficult-to-map regions. That's what it was set up to do. Essentially, DRAGEN has no issue running and analyzing your large-scale sample sets if that's what you typically find yourself dealing with. On the other hand, GATK is the industry standard for SNP and indel discovery in germline DNA and RNA-seq data. It's developed by the Broad Institute and provides a number of open source tools that can be used in order to run your analyses and pipelines. DRAGEN-GATK came about as a partnership between Illumina and the Broad Institute. The DRAGEN-GATK tools were specifically designed to be fast, reproducible, and accurate like DRAGEN, and the DRAGEN-GATK best practices contain open source workflows that are compatible between Illumina's platforms and the normal computer hardware that you're probably already using. So this might actually be something that you're interested in doing because fundamentally, the DRAGEN variant calling pipelines are optimized to run on Illumina's proprietary hardware-accelerated FPGA chips, which you might not have access to. However, DRAGEN-GATK is open source and optimized to run on whatever it is that you currently have available. So I need to make it clear that as a result, DRAGEN does not call variants exactly the same as DRAGEN-GATK. Both Dragon and DRAGEN-GATK run off of completely separate code bases currently under active development. So since the code isn't the same, there is always going to be a certain amount of background noise that exists between them. Our goal with DRAGEN-GATK was not to make the output identical between these two pipelines, but to make it so that the output is statistically indistinguishable from the general background noise that we should expect from running these kinds of analyses. This kind of statistical indistinguishability is what we refer to as functional equivalence, and Michael Gatzen has a presentation prepared for us today on this topic coming up soon. So DRAGEN-GATK can be installed and run from the terminal, just like any of our GATK tools. That means you can run it from many different possible environments and even a few impossible environments if you want to hack it for yourself. But did you know you can also run GATK from the cloud using Terra? Terra is our cloud-native platform for accessing data, running genomic analysis tools, and for collaborating and sharing with others. This makes it very practical for demonstrating pipelines in a webinar environment like this one, so it's a perfect opportunity for you to get your hands on a fully functional DRAGEN-GATK pipeline and use it and modify it as you wish. So just to be clear, you don't need to run Terra in order to run DRAGEN-GATK. However, once you have access to this workspace that we're going to give you access to, you will also have access to all the scripts that it's running, all the inputs that it needs, and that means that it will be very easy for you to move this project off of Terra if that's what you want to do later. And for those of you who already use Terra or you end up liking Terra after this webinar is through, you are also completely free to upload your own data into the DRAGEN-GATK workspace that we've provided to you and test it out using our temporary billing project, which we have also given you access to. Today, I'm going to be walking us through our featured workspace in Terra for performing variant discovery on whole genome germline samples. I'll be showing you how to align an array of unmapped reads that we're going to start off with using the DRAGMAP aligner, and then we'll be calling variants using haplotype caller, which will be running in Dragon mode. This is a pretty powerful variant calling pipeline and one that we're all pretty confident that you should be able to use for your next project. This, incidentally, is what James Emery is going to be presenting about in more detail when I'm done talking here. I'm definitely going to go over this in a lot more detail during the walkthrough, but for now, I'll just let you know that this workspace is freely accessible on the app.terra.bio website. All you have to do is go to featured workspaces where you can search up the key term Dragon, and it'll be the first item that you see. Also, before we move on, I want to remind you all that all of our DRAGEN-GATK resources are available on our website at gatk.brodeinstitute.org. From the main page, we have a link there directing you to our DRAGEN-GATK resources page. And on that page, you will find all of our articles, workspaces, videos, and presentations related to DRAGEN-GATK, even this video. All right. Well, that brings us to the end of the introduction here. So from here, I'm going to pass the mic over to James Emery, who will be talking to us more about the DRAGEN-GATK pipeline and how it works. So James, the floor is yours.
Hello. Thank you, Derek. I am James Emery. I am, as he has already introduced me, I'm the primary author for DRAGEN-GATK. I'm about to give a presentation on just sort of high level what it is, what changes came with it. I will note that this presentation does presume that you have at least some passing familiarity with how haplotype calling works and how our general pipeline works. You know, if you've been to one of our workshops in the past or you're sort of familiar with these tools, you probably already are aware of this stuff, but, you know, be willing to ask questions later on about this if this is too much for you. All right. So this is, so I'm going to give sort of a broad scale overview of what actual changes and what DRAGEN-GATK corresponds to from like a coding perspective. So, you know, if you really get into it, what is Dragon? What, like, you know, how does it relate to GATK in the first place, right? Because obviously, you know, GATK is sort of an old project. We've had variant calling for quite a while. So sort of the history and what Dragon is in relation to us is that, you know, they have a version of our original variant caller from back in the GATK 3 days that they've rewritten basically most of our best practices. They've rewritten it in C, they've rewritten it using FPGA to speed up the very like expensive, slow steps But on top of that, they've also made some scientific improvements. So they have a very fast version of our old GATK3 pipeline that's sort of evolved a bit since then. So a lot of the work that went into making Dragon GATK is sort of bringing GATK up to speed with the improvements that they've made, and also bringing, working with Dragon to make improvements on their end so that everyone is sort of on the same page and we're producing similar results. And so when we get to Dragon GATK, obviously, when you're talking about two different code bases produced by different companies, it's impossible to get everything, exactly matching. Derek has sort of talked about there being this concept of functional equivalence. And sort of just to hammer this point home, right? When we say that these tools are functionally equivalent, we have a version of our pipeline that runs with a bunch of extra settings, it runs a bunch of extra algorithms that produces variant calls from your original input BAM that are functionally equivalent to the variant calls that you would have if you just ran exactly the same BAM through Dragon, just like with FPGA hardware, right? Now, what does that mean? I think I will leave it to Michael, who's going to speak a little bit later, to talk about the actual proper definition of what we mean by functional equivalence. But the idea is just that the discordance for these variants is very low. And just to give you an idea of what we mean when the discordance is very low, right? These are some ROC curves for, on the left, SNPs, and on the right, indels for CHM113, artificial synthetic diploid samples, right? So, they're samples that we have pretty good truth for. The important thing is that you can see that these lines, sort of the orange here being Dragon 3.4.12, which is the version we targeted originally for functional equivalence. The blue being the original GATK pipeline for haplotype caller, and the black here being Dragon GATK. You can see that those curves are basically on top of each other, right? Like when we say that we're functionally equivalent, I just wanted to, people to have the image that we are actually, in terms of ROC curves, in terms of quality metrics, in terms of actual variance called, we are very, very close to matching what Dragon outputs. And, it's not exactly a one-to-one correspondence, but it's very, very close. And we're doing more or less the same things at the same sites. And, generally speaking, this has resulted in a version of, haplotype caller calling and our just general pipeline that performs better, right? Like those ROC curves are pushed more up into the top left of the screen, which means you have fewer false positives for the same sensitivity, which is great. If you want to sort of subdivide regions, if this being the LCR or low complexity region, so if you're looking at sort of the hardest regions, maybe the old version of GATK was arguably better, maybe the story is sort of messier if you go looking for sites where you might be mismatching. But the important thing is that we have a version of GATK that is now very, very close to concordance with Dragon. And so what changes did this require? What do the pipelines look like? I am going to talk about a little bit about what changes to our overall pipelines happened, and sort of what Dragon looks like and what they do. And then I'm going to talk about our new pipeline, and then I'm going to talk about some of the haplotype caller changes and algorithms that have actually become part of Dragon GATK. So this is sort of a high level and somewhat focused on variant calling representation of what the Dragon variant caller does, right? So on the right here, you can see they use their Dragon aligner. They have their own, FPGA accelerated aligner that is, has advantages. It's alt-aware. It does a bunch of extra things. But then there's sort of there's this path on the left here that does where you sort and duplicate mark, you detect active regions, you do assembly, you run your HMM, and then finally you feed those results into the genotyper, right? If, you have a passing awareness of how haplotype caller works, or, have been to any of our previous talks, those steps are basically totally analogous with what haplotype caller, short variant discovery, what our pipeline does right now, right? Most of those steps are folded up into what we just call the haplotype caller, right? But those are all sort of, on the FPGA, you run this all as a continuous pipeline. But in GATK, we sort of run it in separate, discrete paths, but those are all exactly the same steps. There are some differences, however. Note on the right, we have this PCR model training, which is called, auto calibration. That's sort of a little bit different, and I will get into what that is, but that relates to what they call STR, like their STR model code. But, notably the differences to GATK here, this is a very fast implementation. It's all happening on a single processor that is a very beefy processor, and it's using FPGAs to be very, very fast when it does that. You will note a few things if you're sort of reading ahead. We don't have a BQSR here. But generally speaking, like the pipeline, the top level takeaway here is that their overall pipeline is very similar to our overall pipeline. And just to hammer this home a little bit more, this is the proper GATK, legacy GATK pipeline. So if you want to do, joint genotyping or single sample genotyping, these are the steps that you would take, right? So you start here on the left, you map your reads. You typically, the mapping would have been done with BWA in the past. Then you take those reads, you duplicate mark them, you run the BQSR or base quality, or recalibrate the base quality scores. And that gives you your input BAM, your output BAM, right? And then you feed those into call variants, so haplotype caller, and you spit out a GVCF. And from there, maybe you run CNN score variants, maybe you run filter variant trances. So this is what you would do in a single sample pipeline. Maybe you feed those into some higher level analysis, and then you do what you're going to do. Run Funcatator, run any number of other tools to take those variants in analysis. So we now have a new pipeline that is running that you can just sort of slot in and run in exactly the same way on Cromwell and Terra like you would have run this pipeline before. That is the DRAGEN-GATK pipeline. So we've sort of adapted that previous pipeline into our new, our new best practices pipeline for single sample variant calling. So let's go through each of the steps. So at the pre-processing step, some things have changed. The big one is that we are now using the DRAGMAP aligner. So no longer is in our standard best practices pipeline are we using BWA. Now we've switched over to using DRAGMAP. They have an open source implementation that can be run in the cloud that works reasonably well and produces fairly concordant results with the actual Dragon aligner. And that gives you all the advantages that they have in terms of, alt-aware assembly improvements and whatnot. So we're producing very similar reads just sort of as the input to all of these other pipelines. Furthermore, we run regular mark duplicates. The duplicate marking algorithm between the two tools is not so terribly different. However, we skip what the previous step that we had, which was BQSR. We no longer recalibrate the scores. We rely on the original aligner quality scores on the reads. Then when we actually get to the variant discovery step, we actually have changed these steps a little bit, right? Now we have this calibrate dragster model. This is exactly the same algorithm that we talked about that sort of was on the right side of that previous slide. This is where, so instead of doing BQSR, you do this auto calibration step where you're sort of learning information about STRs. And I will get into that. That is in future slides. But then that goes into the haplotype caller in Dragon mode, which is sort of the new version of haplotype caller that has all of the extra algorithms and improvements that Dragon have, that the folks at Illumina have come up with. Finally, once you have your call set refinement, typically as part of our best practices pipeline, we actually don't run CNN score variants. We don't do any sort of post-mortem filtering. We rely on the quality scores for Dragon for the DRAGEN-GATK module being much more accurate, and those quality scores can actually, at least in our basic version of this pipeline and our best practices version of this pipeline are actually good enough that we don't have to worry about doing any sort of post filtering. Like you can just take the VCF that comes off of the haplotype caller and use that. But there's nothing preventing you from running VQSR or anything after that. It's just for the single sample pipeline, we consider the qual scores to be good enough, and that is exactly in line with what Dragon do themselves. Cool. So let's get into the actual error models and what the actual code improvements that come with DRAGEN-GATK are. So I've talked about it repeatedly, but there's sort of first there's the STR model. So STRs are, stand for short tandem repeats. They're sections of the genome where you have lots of little repetitive units. There is a new pre-processing model that, as you've seen in the previous slides, there's all these extra steps into the pipelines where you're sort of in tandem with actual variant calling, you're learning some detail about on a per sample basis about STRs. And those are fed into the HMM. There's also two other primary genotyping algorithms that are improvements to regular GATK that have been ported over from DRAGEN. Those are base quality dropout and foreign read detection. So base quality dropout, and I will go into more detail about these two, I promise, is sort of like a model for context-sensitive linked base quality errors. So it's sort of an extra model that lives on top of the genotyper that tries to suss out like, oh, maybe this variant comes from, an artifact of the sequencer. And so we sort of want to repair those errors. The other model is sort of a grab bag that handles a bunch of different edge cases. This is foreign read detection. So it is sort of a model that attempts to, in the genotyper, when you're actually generating your scores, it attempts to sort of to compensate for all kinds of things. It handles mapping artifacts, it handles potential contamination. And both of these models have the effect where you can sort of recover sensitivity and sort of do a better job at genotyping, so you need less filtering because you're just producing more accurate results at the level of genotyping. And both of these have the net result of reducing false positives, which lets you sort of like emit more marginal sites with knowledge that you aren't emitting too many false positives. All right, now let's get into STRs. Just a reminder, what are STRs? STRs are sort of repetitive sections of the genome. These are all over the place in the reference, where you might have the same pattern repeated over and over again, right? So at the top here, right, you might have a TC, 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 right? And that would be an STR of periodicity 2 that is repeated 5 times, right? And at the bottom, it might also be TTG, TTG, right? That's a periodicity 4 with a repeat length of 2, right? So what does STR do, actually? Well, what it effectively, like, STRE model that we've ported from DRAGEN, it is a model that attempts to, on a per sample basis, so not universal to all sequencing technology, but universal to the actual like individual sample that's come off the sequencer, we try to learn a prior on how likely it was at any given type of STR. Was there an error? Or how likely is it, or how confident are we that if the read shows that there were actually 5 repeats of this element, there were really 5 repeats of this element? And so, if you want to get super into the weeds of how old GATK worked, deep in the HMM, in the pair HMM that we use to actually generate like haplotype to read quality scores, those are likelihoods. Those already had baked into them a model for indel errors that was sort of based on repetitiveness. This is being much more, going through the results with a much more fine-toothed comb. So now we're saying, not only do we care if we're in a repetitive region and we're going to lower the probability and our confidence that a particular read is accurate, we're also going to lower it based on exactly what type of STR we see, right? So these two different STRs here are actually going to be represented in our models and result in different priors that we're using for HMM generation. And so we're generating big tables where we've sort of pre-computed by subsampling across the whole genome. Oh, at all the sites where there were 4 length STRs, how likely was it that we saw errors? And we sort of use those to generate a model and sort of like adjust the scores that then go into the genotyper and generate and ultimately generate more accurate likelihoods for our reads. So here's the math. If you really care about it, there's like a model that we're learning. We're sort of subsampling. Ultimately, this old math gap penalty calculation that we were using in GATK, that's basically just looking at the repeat length and doing some math there that is sort of flat, right? So if you have, a 4 base STR that is repeated twice. That's a repeat length of 8, right? In the old model, we treat that exactly the same as a 2-base STR that's repeated 4 times, right? Like exactly this previous slide. And so we sort of don't have any, we can't disentangle those two and give a more accurate genotyping for one without hurting the other. With the new model, we sort of learn, and don't worry too much about the math. With the new model, we are learning a table that is able to distinguish those two and generate an empirical likelihood score that's used as a prior for both of those two models independently. And so we end up with much more accurate HMM error computations, which then feeds into the genotyper and results in overall better genotyping, particularly at sites where you have lots of indels. All right, and so that's just sort of a high level of how STR works. This is sort of a separate tool where you take in your BAM, take in a list of STR sites that you know for your particular reference. You subsample and do some math to sort of figure out a model of how likely for that given sample and any given indel error is to occur, and how accurate any given read is about that particular type of indel. And then you feed those into a table, and those tables ultimately get used in the pair HMM to produce better likelihood scores. That's STRE. That's the first one of the major model improvements that came with DRAGEN. The next ones are sort of, the next one I'm going to talk about is BQD. So the sort of underlying logic of BQD is that there's sort of an understanding that for certain sequencing technologies, be those Illumina, be anything really, that there are maybe contexts, like sort of at its core, right, when we genotype, we have the sort of, we make the assumption that everything is that errors are independent. And that's not really necessarily true, right? Maybe there's certain reference contexts that cause all of your reads in the sequencer via the same technology to start to sort of generate lots and lots of errors because that context, just for whatever reason, breaks the chemistry. This is a model that attempts to fix that. So if we see here, this is just sort of a version of IGV. And you can see on a per strand basis, there are lots of reads here that have these C errors, right? But if you look at the base qualities, they also totally drop out. There's lots and lots of errors in all of your reads after a certain point, but there's enough reads that all sort of have the same exact error in one place that maybe it gives you reason to consider that this is real. However, however, our current old legacy genotyping model can't really handle these. It's going to see, oh, we have a couple of reads that all support the same haplotype, that all have the same variant. We're going to call that very confidently because it does look like a hat, whereas it might not actually be a hat. It might be that you've seen some artifact that's like this. So that's what base quality dropout aims to fix. It is a genotyping model. You are sort of learning a threshold. It is sort of an extra genotyping model on top of the existing genotyping model that we use for diploid sampling, right? So we're saying, instead of generating, for a single variant, generating the likelihoods, and then calling a genotype for, the sample being homozygous reference, heterozygous reference alt, and then homozygous alt, right? Instead, you are also computing, well, what is the likelihood that, it was homozygous reference and all of this other stuff that I see is just a result of this base quality dropout, like one of these artifacts, right? Or what is the chance that the sample was homozygous alt and the reference reads were all because of this artifact, right? And so that's sort of what we're looking at. And generally speaking, this is a model, it's learning a threshold and then doing genotyping calling and it's spitting out the best result based on a bunch of factors, mostly the base qualities, the read strandedness, the cycle count for the particular base that corresponds to the variant that we're looking at. And importantly, this model also, at least currently in GATK, only applies to SNPs. And I believe that's also true, at least in some versions of DRAGEN as well. So just to sort of go into what the math at the bottom means, I should note that, it is actually not so terribly different than our existing likelihood score. And if you've seen our other talks, you've actually seen this math before. You can see in the middle here, there's the probability of a read for a given genotype over 2, right? That's just the diploid calling math that we typically use, right? So, and those just come, we get those scores directly from the HMM. But it's sort of repeated twice. There's this error term, you're summing over a few things. So I should just note, like, the way the model actually works sort of at its core is we're sort of sorting all of the reads by their position and by their strandedness, right? And then we are sort of choosing a threshold based on the position and based on the base quality to sort of find a cutoff for each of the strands where we say every read after this point we think is probably actually error, right? Or there's a reasonably high chance that every read after this point is probably really error. And that's sort of what those error terms on the right are, right? So on the left here is the normal genotyping score for all of those reads that existed before. That would be just what we calculate in regular old legacy GATK. And everything on the right side of this has this extra error term modifying their probability. And those are everything that's sort of below that base quality line that we have here. And so that sort of lets us generate a new model where we pick the maximum across this whole calculation, where we say, okay, at this cutoff, like, you know, if we sort of assume all of these reads are towards the end are error, well, that ends up with us getting a slightly lower quality score, and that lets us sort of pick out errors, right? We're sort of penalizing situations where you can cleanly pull these error terms out. So that's BQD. That just lives in the genotyper. It results in sort of new likelihood scores or new PL scores for all of your reads, and ultimately better genotyping and emitted variance from a haplotype caller. The next model is foreign read detection. Just to sort of give you a high-level overview of where, you know, what foreign read detection tries to pick up, right? This on the left here is another sort of modified IGV screenshot. This is an example of one of these like potential sites that they want to that FRD sort of aims to highlight. So if you can see here, there's all of these reads that are pretty well phased that look pretty accurate, that there is a T variant, a T SNP here, right? However, you can see there's this other family of reads that are very well phased with each other that don't have the T SNP, but have two Gs next to each other, right? And this sort of gets at one of the advantages of doing the genotyping model in this way, the way that Illumina has chosen to, that we've ported, is that, you know, you can see here, if you aren't careful, you would probably end up calling this as a heterozygous site for the Ts, right? But that's not real, right? There's a lot of evidence here, if you actually look at the variants, that, oh, well, those Gs are all phased, and, you know, in reality, you can't see this on the slide, but they also happen to have much lower mapping quality than all the other all the other reads at this site. The GG is localized. Maybe there's something going on here. Is it possible that this is actually a mapping artifact, or that this is actually sample contamination? Is it possible that any of those things are causing this to look like a real alternate allele at this site? But how do we do that? Well, I'm not going to go into the math for FRD. It's a little bit more complicated than the BQD math, but the basic premise is the same, right? You split the reads by sample. You try to penalize anything that's not sort of relatively, like if you're going to make a het call, you're sort of penalizing it if it doesn't look like it's sort of close to 50% or close to your diploid, your ploidy like percentages. It also looks at things like mapping quality. It looks like, again, strand bias. It attempts to, and it ultimately attempts to make a model that works very much like the BQD model before, where you're sort of generating a cutoff and then you're penalizing variant calls that are at, that would result in het calls that are not real. So both of these models are sort of ultimately depressing like the ability for you to call hets, and making you much less confident about het calls in places where there are these artifacts where you can cleanly divide the reads by mapping quality, for example. Cool. All right, so that's BQD. There's a few other minor things to talk about in terms of what, you know, this is less, you know, in the weeds of how haplotype caller and dragon work. This is much more of user-facing. This is a little bit more of a user-facing thing. But one of the big differences that happened between Dragon and GATK is that the call scores are now wildly different, right? In GATK, it wasn't terribly uncommon if you look at the right here. For the same variant site, for a variant site to have a qual score, which is ultimately like a Fred score that's computed based off of the PL array, the GL arrays that we've computed, in the range of like 3,000 and, you know, like upwards into the thousands, right? Particularly for homozygous sites where you're very confident and you see like, you know, dozens of reads that all have the same SNP, for example. That is not very real, I guess, in a word. Ultimately, at a site like that, like, you know, if you actually think about what how the calculation for Fred's quality works, right? Being 3,000 confident means that you're effectively saying that you are 1 to the 300. That you expect the error happens on the order of less than 1 to the 300, right? Which is absurd. Like you aren't that confident. You aren't that confident in your technology, your reads, your alignment, any of that. So it's all been sort of like, you know, when we go to filter, part of the GATK paradigm generally has been that for when we worry about quality scores, it's sort of as just another quality that gets sort of fed into annotation filtering, like BQSR or like VQSR, like all of our typical filtering algorithms. And we sort of don't care too much about the original quality because it's sort of artificial. Well, if you're dealing with Dragon data or Dragon GATK data, there's actually a pretty substantial difference in how the quality scores are computed because of those previous two genotyping algorithms that sort of depress and penalize heterozygous calls. Ultimately, you end up with, well, the heterozygous calls by design end up getting capped at 50 qual anyway, right? So you're going to have this huge spike of scores that all line up on this 50 number. And those are basically all of the hets or all of the confident hets that you normally would have called in the range of hundreds or thousands of qual score. Those are now lower, and those are all capped. And then, you know, the qual scores are sort of now lower. And this is important for people to know if you're planning on using Dragon GATK or Dragon, that, you know, the scores are the quals are very different, and you have to be careful when you're sort of mixing and matching data with older legacy pipelines, that the pattern sort of looks different. And there's a few other annotations that might also be different. Then, you know, this is just getting a little bit back into the weeds, but, you know, generally speaking, there's some changes also in the, another algorithm that's been ported from Dragon is this dynamic redisqualification. It's just a kind of a tweak, I would say, but it does mean that, you know, just sort of the general pattern with Dragon is that because you have the better variant calling models or the better genotyping models for FRD and BQD, it means that you can sort of rescue more reads that actually make it to genotyping. And so this model is just, you know, it means that you're being much more conservative about what reads you throw away because they probably are foreign. And ultimately, then the genotyper is better able to compute with that. So part of the reason that Dragon does perform better is because the net result of all of these extra algorithms is that you can just use more of your depth. You know, if you have lots of low mapping quality reads, maybe you can still make a call or at least use those reads as evidence for or against a given allele, which is ultimately what we do and what this sort of dynamic redisqualification results in. All right. I know that was a lot. That was a lot of technical stuff. I'll be around for questions later if any come up, but thank you for your attention.
Excellent. Thank you so much, James. I really appreciate how you broke it all down for us. So like I said, we are going to have a Q&A session after the demonstration. So if you have any questions that you just thought of, please go ahead and post them as they come to you in the chat. We will address all of them in that session. All right, so coming up next, we have Michael Gatzen. He's going to be presenting to us on functional equivalence so that we can actually get more information about how Dragon compares to Dragon GATK. Michael?
Yeah, so yeah, I am also working in the data science platform. and I've been doing some work on evaluating functional equivalence. And this is usually in the topic of benchmarking. So maybe to take a step back, when we think about benchmarking, we usually think about beating the competition. So we have two pipelines, we run them both, and then we look at the VCFs, compare them against truth, and then look at metrics like sensitivity and precision to then determine, okay, this pipeline is better than the other pipeline. As studies become larger, though, data sets are more useful when they're combined from different sources. So different sequencing centers, for example, want to all sequence the data and then combine them. And so we need to combine the results from different methods, tools, or pipelines into larger biobanks or data sets. This, however, introduces the risk of batch effects, which means that there is slight differences in the results of the data depending on which tools they have been called on. So the question really is, how can we ensure that data from different sources can be used interchangeably and without batch effects? Now, we can't expect the results from two pipelines to be byte equivalent, but we can talk about this concept of functional equivalence. And in this case, of course, we're interested in the functional equivalence between DRAGEN and DRAGEN-GATK. How do we determine functional equivalence? So this is usually done by taking a sample and producing two independent replicates of that sample. So sequence it twice and then run both tools on each of these replicates. And then we can compare the output of the different tools run on the same replicate, which is what we call the inter-tool difference. And we compare that difference with the difference between the replicates called with the same tool, which is what we call the intra-tool difference. And if we then find that the inter-tool difference, which is the bias due to the difference in tools, is smaller than the intra-tool difference, which is just the noise inherent to genomic data analysis, then we can say that these tools are functionally equivalent. How do we quantify these differences, though, these differences between call sets? So we need to look at the concordance between the two call sets, which is just the overlap of calls between the two VCFs, basically, as well as the difference in quality. None of these criteria catches the difference alone, so we need to look at both. For concordance, we can just define one sample arbitrarily as truth and the other one as an evaluation sample, then run a tool like VCF-eval and compute the Jaccard score, which gives a measure of how many calls are shared between the two files. We can then plot the results in a plot like this, where we, let me just move the Zoom windows away. In a plot like this, where in the middle we have the Jaccard score between the two tools, one on the same replicate, and then on the sides we have the differences between the replicates with the same tools. And if we find that the Jaccard score for the inter-tool difference is larger, so it has a higher concordance than the intra-tool difference, then we say that this criteria is fulfilled. We do this for certain regions in the genome and for SNPs and indels. And then if all of these plots satisfy the condition, then we say that the concordance criteria is valid. Now, as I said, we also need to look at the difference in quality. And some of you might be familiar with the concept of ROC plots. I'm not going to go too much into depth. But this is what we usually use to quantify quality differences. However, all these ROC plots don't capture all the information either, because we can have, like in this case, two very similar looking ROC plots, but the points on the plots on the different curves can widely differ. So if this point corresponds to Q20, and this other point corresponds to Q20, and then we obviously don't have an equivalent performance or quality of the course. So for that case, we use these so-called F1 plots, which, again, I'm not going to go into detail. All of that is described online in various articles. But basically, we plot the inter and the intra-tool difference in the F1 scores along an axis of different quality thresholds. And then if we find that the inter-tool difference is smaller than the intra-tool difference, then we find that this criterion is also fulfilled. This also we do on different regions in the genome. So we have these plots for the concordance side and for the difference in quality side. So really this whole concept and this workflow for functional equivalence combines or makes a three-way comparison between the VCFs of the different pipelines, as well as a truth sample that is provided. This is probably the most interesting slide, which is the configurations that we need to use on both sides of DRAGEN and DRAGEN-GATK. On the DRAGEN side, we need to use different settings, and all of them are described in this article called Functional Equivalence in DRAGEN-GATK. Maybe we can post that in the chat. And on the GATK side, if you're running the GATK with the warp pipeline, that the WDL pipelines that we provide, then there is an argument that says DRAGEN functional equivalence mode. And if you set that to true, then automatically all the settings are used correctly that you need to produce functional equivalent results on the DRAGEN-GATK side. This workflow, as well as everything else, is on GitHub using this link. There's also a featured workspace, a featured Terra workspace, that you can copy and then experiment with the concept of functional equivalence, use all your own samples, and make sure that you also find the two pipelines to be functional equivalent. And as I said, we will be posting the article that describes the different settings and the different configurations that you need to use to produce functional equivalent results between DRAGEN and DRAGEN-GATK.
Excellent. Thank you so much, Michael. For the next part in our webinar, what I'll be doing is actually walking us through the Terra workspace. So why don't I go ahead and share my screen? Great. So this is going to be a walkthrough and demonstration. So I encourage you all to actually follow along with what I'm doing here. And that's going to involve you logging into Terra and finding the workspace for yourself. There should be a link to the workspace in the chat or in the YouTube description, but I'll show you how to get it here. First off, why don't we go to app.terra.bio. That's the first place we need to go. So we can actually go to Terra. I'm already logged in, but if you're not, there should be a login button here where you can log in. So the next thing that we need to do is find our workspace. So fortunately, this is a featured workspace that we're going to be showing. So if you click on the library button here and you go down to featured workspace, then you'll click here and you will be taken to the featured workspaces section where we have lots of different workspaces that you can use in Terra, everything from copy number variants, germline preprocessing, variant calling, and anything. But for now, we're going to be looking for our DRAGEN workspace. So up here at the search menu, you just look for DRAGEN, and it's the first item that pops up. This DRAGEN-GATK whole genome germline pipeline workspace. So we'll click on this here. So the first thing that we're going to look at here is the dashboard. The dashboard of this workspace has all the information that you need to know how to run and modify this workspace, along with links to many different resources that you can use to get a better understanding of how this pipeline is being run. However, the first thing that we're going to notice here is that this workspace actually doesn't belong to me. It says this here in the corner. It says workspace is read only. You should see the same thing as I do. And if we come here to the workspace information tab here, if I click on this, we'll see that my access level is reader. So I'm not actually an owner to this. That means I can't run this workspace and I can't edit this workspace if I want to. If I come here into the owners section, also my information is not here. These are the people who actually created the workspace on Terra. So the first thing that we need to do is make a copy of this workspace on our account so that we can access it. So that's going to involve coming up here in the top right corner. There's a button here, a 3-dot options button. If we click on this, we'll get more options. And there's an item here for cloning the workspace. So if we just click on this clone workspace button, then we'll get an option to make a copy of this for ourselves. So the first thing I'm going to do, so I don't get confused, is change the name of this to something that I can find later. I'll put my initials and there so I can differentiate it. And the next thing we're going to do is select which billing project we actually want to have connected to this workspace. If you did all the steps and were given access to the temporary billing project that we provided to you for this workspace, then you can easily find it in workshop-GATK-2210. That's one of the options here. So I would recommend that you do this so that then when we run this workspace and download our files, you won't be charged for that. We will be charged for that. You can leave the bucket location as the default for now. And once you've changed the name and added your billing project, then we can just click on clone and we will be given a duplicate of this. And there we go. We can verify that this workspace is now belonging to me because up here in the corner or in the top, you can see that it's got my new title that I gave it. And in the workspace information section, you can see that my access level is now project owner. That's different from before where I was a reader and couldn't do anything. And here, if I click on the owners tab, then I can see that I am now a owner. That's my email address. Another thing you can see, we'll have access to be able to edit this. If you want to edit this later and share it with your colleagues, for example, then that's how you can do it. There's options here so you can make this workspace your own. So what I'm going to do now is give you a little bit of a tour of the workspace itself. If you're not familiar with Terra, this will be a good opportunity for you to see the different options here and also how we can take all this information off of Terra when we're done with it. So the first thing I want to show you is on this dashboard. I want to show you how this workspace is actually going to run. We've already covered this in the previous presentations, but as you can see from this diagram, what we're actually going to be doing is inputting unmapped reads here, and we're going to be mapping that to the reference using DRAGMAP aligner. And then we're going to be calling variants using haplotype caller that's going to be set in dragon mode. And from that, we're going to be given some VCF files that we can use that will have all of those variants written into them. So if we scroll down a little bit here, we will go to the running the workflow section of this dashboard. And there you will have information about the two different workspaces that we actually have access to in this workspace, or sorry, the two different workflows that we have. The first is this functional equivalence mode, and the second is this maximum quality mode. So the functional equivalence mode workflow is going to produce outputs that are functionally equivalent to the same inputs being put into the Dragon hardware. So the purpose of this workspace is to make it as functionally equivalent with Illumina's hardware as possible. The second mode produces outputs that are with the maximum quality that's possible. So it's going to be performing additional steps like unmapping contaminant reads or using spanning event genotyping. And that is going to produce these outputs that are of the maximum quality possible. However, this is going to come at the expense of the outputs not being as functionally equivalent with Dragon hardware. So I would recommend that if you are currently using Illumina's Dragon, or you think that you may be using Illumina's Dragon in the future, that you stick with the functional equivalence mode. If that is not something that interests you, then I would recommend the maximum quality mode. So if we go back up here to the top, and we visit the data page, you should see a button up here for the data page. We'll move over there. And this is where all of our sample data that we'll be using to run the sample workflow will live. And of course, you can substitute any of this example data with your own data later on. But just so you know, this is where it's going to be housed. So we have three options here. We've got tables, reference data, and other data. If we take this one at a time, we'll see that in the tables section, we've got these read groups. So if I click on here, we'll see that in the read group section, we have all of our unmapped bands. So I'm actually going to expand this out so you can see these are all unmapped bands, which is the input that we're going to need for DRAGEN-GATK in this workspace. Next, we have this read group set. And so this is going to organize all of the read groups into a single set belonging to this NA12878 sample. And later on, we're going to, when we get our outputs, we're going to find all of that information, all the metrics, and all the VCFs housed in here as well. And down below here, in the reference data section, if we click on here, then this is going to have all of our reference and metadata. That's going to include our index files, interval lists, and anything else that we want to point our pipeline towards. There's going to be a lot more information in the documentation that I'll be sharing with you about what these are and what you need if you want to replace these with your own data. And here in the other data section, we can click on this. And you'll see this is just additional workspace data that the workspace can have access to. For example, here's a haplotype database. If you click on any of these, by the way, you can download it and perhaps even get a preview of what's in there. So I'll click done. And OK, so now why don't we move on from the data page? Let's move over to the workflows page so that we can actually see the two workflows that we have. So now that we're here, we have these two workflows. The first one, like I said, is this functional equivalence mode workflow, and the second is this maximum quality workflow. And so again, the functional equivalence, we're going to be running this today, but this is going to give you outputs that are as functionally equivalent as they could be with DRAGEN hardware, whereas the maximum quality mode is going to give you just the maximum quality possible, regardless of how functionally equivalent it is with DRAGEN. So if we click on this, we'll be taken to the workflow setup page, which is going to have relevant information about the workflow, like the name, the version. So here's the name, the version you can select here from this drop-down menu, and also the source on GitHub for all the code that's going to be used in this workflow. Actually, if we click on this right now, we'll click on it now. And I encourage you to click as well so that you can see this. We will open up a new tab, and this is going to give us the source link, and it's going to take us to DocStore. So in here in the DocStore, we will have all the information about the script that's going to be run for this DRAGEN-GATK pipeline. So I'll also say, for those of you who are interested in running this DRAGEN-GATK pipeline off of Terra, there are options here for you to launch it in DNA Nexus or Anvil or whatever you want. And also, if you click here in Files, you can see the actual raw source code that you can use. This is all written in WDL, WDL. It's a workflow description language. So if you have experience with that, you can modify this code as you want and use it. You can download it here as well. So I am currently interested in showing you this on Terra, so I will close out of this and go back. And I'll just show you here, we have three options here for script, inputs, and outputs in the actual workflows page. And if we click on the script, you'll see this is the same script that we were looking at before. This is what is actually going to be run in this workflow. However, here in the inputs tab, this is going to be all of our inputs. So all of our inputs here are already configured for us, and they're going to be pulling sample reference information from the different data tables that we were previously looking at in the data page, okay? And here in the outputs, it's going to be the same thing. These are going to be all the different metrics and files that are going to be generated. These are going to get written into the outputs. These are going to be written as outputs back into these read tables here in the data page. So if you're going to be editing this for yourself, for your own data, then you will probably want to look here to make sure that the information is pointing towards the right data that you want it to. Okay, and before we get started, the last thing I want to show you is this job history page. Once we run this workspace, then we will be able to verify how it's being run. We'll be able to monitor its progress in this job history page. Right now, we're not running anything. We're still on the tour. So this is empty. There's nothing here. But okay, so why don't we get started? Up until now, we've just been taking a tour of the workspaces. So why don't we take all these elements that we've laid out here and we'll put in a bowl, we'll mix it up, and we'll bake ourselves a nice little DRAGEN-GATK cake. So yes, let's go to the workflows page and visit the functional equivalence page again, one more time. And this time we're actually going to try and run it. So in order to do this in Terra, it's very easy. You just need to follow the steps. Step 1, step 2. Okay, for step 1, why don't we select our root entity type? This is already set for me as read group set. So that's, again, the file that's going to have all of our outputs. And then the second thing we want is step 2, select data. So when we click on here, we're going to select the specific read group sets that we want to process. And we only have one selected here. We'll click on this checkbox. And that's going to be available in the data page as we saw, so that we can select it and then push OK. So why don't we do that right now? And then when we're done, we're going to click on Run Analysis. You'll be given this pop-up window here. So there's nothing for us to do here unless you want to add a description of your submission, but we can just click Launch and it will run. And there we go. Now we are taken automatically to this job history page. And unlike before, you'll see that we can actually monitor the status of our run. It's for right now, it's queued for me, as it should be for you. And after the workflow finishes processing, all of our outputs, like our VCF file containing our initial variant calls, will be written back into here into the data page, into this regroup set table. So it's not going to do anything now because it's still processing, but we should check back in on this later. So while we're waiting, why don't we go back to the dashboard, where again, we have all of our information here about this workspace. And I'm just going to show you where you can find relevant information about the workspace later. If we scroll down to the workflow overview section right here, you'll see that we have a lot of information here and links. So here it says read more about the workflow modes, inputs, and tasks in the WGS overview. If we click on this, as I am doing right now, we'll be taken to a GitHub repository with all of the warp documentation for this workflow. And this has all the information that you could possibly want about how this workflow is being run, what the inputs mean. Actually, I'll scroll down and you can see what the inputs are, what descriptions of what they do. And this is what you'll need to know in order to edit this workspace on your own. Also, there's information about different arguments that you can use to modify the workflow and the DRAGEN-GATK for your own needs. So speaking of modifying things for your own needs, I'm going to close out of this, and then we'll go down to the customizing the workflow for your data section. And this section has information about where to add in custom inputs, where to download input JSON files from the workflow configuration page, and also how to change it for your own needs. So why don't we go ahead and check back in on the job history. And it's still submitted there. The next step, it's going to be running. And then after that, we're looking for a check mark. It will turn green and let us know that's done. If you want an idea, why don't we return to the dashboard very quickly? Towards the bottom, if you want an idea of how long this is going to take, we have time and cost estimates. So here for about 24 entities running the functional equivalence mode, it says we're going to take about 4 hours for 3 GB file size, and it's only going to cost less than $1. So again, we're paying for that if you're using our billing project, but just so you know for your own data, this is a rough estimate of how much and how long it's going to take. Fortunately, I'm not going to hold you hostage here for 4 hours while our samples are processing. So what I'll do is just show you a previous version of this that I ran yesterday. And this is it's the same workspace, but you'll see that here in the regroups page, we actually have like output VCFs that we can look at, as well as all of these metrics have been populated. If you want to download any of these, you just need to click on them, and there will be a download link that you can use. And of course, you don't actually need to run this in Terra if you don't want to. Like I said, you can go to the doc store, you can run it in your own platform of choice, or you can go ahead and just take the whole thing off of Terra and run it onto your computer locally. But for now, that's what we're doing. You should all by now have access to the Terra workspace, and your sample should be running. If you want, just leave it there on the side. You can close out of it and return to it later. It'll still be processing without you looking at it, but that's it for the demo. So now why don't we move on to our Q&A session, and I'll go ahead and take a look at the questions that you've been posting, and we can address them. So the first question that we have here is, can we use analysis-ready reads generated by the DRAGMAP aligner for somatic variant calling? So yeah, right now what we've been doing is looking at this whole genome Germline pipeline. So the question then is, can we actually use it for somatic variant calling?
Yeah, so taking the DRAGMAP output for somatic sounds, there shouldn't be any particular issue with that. It is more or less slot in and produces results that are on par with BWA. However, it is alt-aware, so it's doing a slightly better job of handling mapping artifacts and issues that come up where there are alt contigs. So we generally see that it just, it produces better results than the existing BWA pipeline.
Excellent. Another question that we have here is, can PacBio reads be analyzed using DRAGEN-GATK? I don't have experience with PacBio, but if the inputs are the same, does that mean that it will also be run?
That is a very good question. I think like just with typical haplotype caller, you can run long reads through it and it works okay. I don't know if we put a lot of effort and energy into making it work as good as it can. I am not sure how the DRAGEN genotyping models would work with PacBio data. It would not surprise me if, because these models, there are some advantages to them, but they are a little bit, in particular, like FRD, in particular BQD, and to a lesser extent FRD, are sort of married to Illumina data and to the error modes of Illumina data to an extent. So I'm not sure how much of a benefit you would see, or even if you would see any benefit.
Geraldine here. So we have a team that has been doing some work with PacBio and developing pipelines that are specific for PacBio. We haven't done a lot of outreach about them yet because they've been in development, but that's something you can stay tuned to, I think. There will be some developments coming soon. And that's not strictly under the aegis of GATK. So that's why it's not entirely integrated into outreach at this time. But there is a group in the wider methods development department that has some really interesting work happening for PacBio data.
And along the same line, since we're talking about the different type of data that we can use, can DRAGEN-GATK be used for calling variants from RNA-seq data, or are we limited in that regard?
As I say, this is more off-label applications that we haven't tested. So, use at your own risk. Our RNA-seq data, it sort of depends on, so FRD and BQD, again, like they are both sort of generic. However, they might allow you to pick up on weird error modes that happen that are sort of context dependent. Like the idea behind BQD in particular is that, there are certain technology specific contexts that cause the technology to sort of fall over, right? Whether that's with Illumina, whether that's with PacBio, long reads are sort of a different beast, and I'm not sure if like cycle count which is one of the things that goes into BQD. I'm not sure if that makes sense in the PacBio case, but for RNA-seq data, that especially if it ultimately is being read in DNA, it would probably be fine. I don't see a reason why it shouldn't work.
Mm-hmm. And I'm assuming that's the same for exome data as well.
Oh yeah, exome should be totally fine. We do, part of our FE evaluations have included exomes.
Excellent. So I'll move on to our next question. We've got a question here. Is there a Docker version of the DRAGEN-GATK workflow available? Yes, I showed it. This question was made before I did the demo, but yes, you can go to the doc store and you can find our version there as well. Next question. How can we share the workflow to use in Terra without group information on my billing project? That's a good question. You can definitely share the workflow by clicking on in the options button, those little three dots where we cloned the workspace. There was an option for sharing, so you can share that with other people. I believe that you need to set up your particular billing at that moment. Geraldine, would you like to weigh in?
Sure. So basically, there's multiple sharing options, but without going into the nitty-gritty, basically you can share. If you want to share your Terra workspace, you can share it with different degrees of permission access. So you can share it just as a reader, so you can allow somebody to see the contents of your workspace, but not actually do anything in your workspace. Or you can share as a writer, and so you can edit things in the workspace, and crucially, you can toggle on and off the can compute property, which means you can give somebody access to actually run work in your workspace, but then if they incur costs, you're going to be charged. But if you disable the can compute access, then they can interact with your workspace, but not actually launch anything that's going to cost you money. If they want to run something, and you don't want to pay for it, then they will need to clone the workspace under their own billing project. And Google offers free credits for people who want to try it out. So people can try this out without actually having to put any money on the table up front.
Mm-hmm. And along the same lines, we have a question here about cost. It says, is it the same cost if my samples are analyzed in DRAGEN-GATK from my terminal as it would be on Terra? And I think the good news is if you're running it on your terminal, on your own computer, then it's going to be free. I mean, you're paying for that electricity. Whereas if you're going to be running this on the cloud, then you're going to be accruing cloud computing costs, which is why having a billing project is important. But if you have access to your data and you have access to a machine to run it on, then just like the rest of GATK, it's totally free.
Yeah, the big thing to consider is what kind of infrastructure do you have access to locally, right? If it's just your laptop, I can say with confidence, you're not going to be effectively able to analyze whole genomes on just a regular laptop, because it requires more computing power and more time than you can have through a laptop. But if your institution has a local, on-prem compute server or cluster or something like that, then you can certainly run it locally. In that case, you'll want to set up the Cromwell execution engine, which is what's in Terra. It's open source. You can install it locally, have your system administrator install it on your local local infrastructure, so you can run these pipelines with a few tweaks. So you can ask in the forum if you're looking to adapt the pipelines to run on-prem, but you'll have to deal with things like queues and so on. So that's where you might want to weigh the options of running locally or on the cloud.
And just a reminder, if you do have any questions that come up after this Q&A, please visit us on the forum. Like I said, we have a DRAGEN-GATK webinar discussion board there, and if you go there, we'll be looking at that going forward so we can answer the rest of your questions. Okay, so why don't we go on? I have a Terra-related, workspace-related question here. It says, how can I create a read group set from the read group table? So actually on the workspace that I was showing with you, it's already set up in the workspace. So it's already been pre-configured and you don't have to do anything to have that read group set generated. So we have the read group with all of our files inside of it in that data page. And then once there's also the read group data table that is going to, the read group set, which is there as well. And it's going to automatically populate that because we've already pre-configured everything.
Yeah, so it's everything that you need to be able to run the demo yourself is in there. If you decide to try this out on your own data, you'll need to upload your data to the cloud and then basically replicate the way the data is described in the data table. You'll have to do that with your own data. Now there's a whole bunch of documentation and examples and videos in the Terra documentation that explains how to do that. And if you're interested, there is, I think next week there's another hands-on interactive Terra session. So if you want to join that, you'll get hands-on training to actually learn how to use data tables in Terra, etc. The idea of data tables in Terra is really to make it a lot easier to be able to run, say you have, I don't know, 300 samples. You get that data table in there. It's basically your data manifest. And then you can select any subsets of your samples, or like all the samples, and just in a few clicks say, I want to run this workflow on all the samples in the same way. And so you configure it once, and then it's kind of, you click it and forget it until it's done. That's the idea. It's a little bit of a learning curve the first time you encounter them, because it's a little different from how things are done when you're working through the terminal. But it's very much worth getting used to, because then after that, anytime you have new samples, add them to the table, a couple of clicks, and you can execute. So that's the advantage.
Mm-hmm. And to be clear, we have a lot of documentation about how to use Terra in more detail on our support website. That's at support.terra.bio. And so there's lots of articles. We have videos on a YouTube channel as well that walk you through how to use data tables in more detail. So if that's something that you want to do on Terra, then we have the resources for you to do that available. If you want to do that off of your machine, like Geraldine said, or off of Terra and off the cloud, then yes, you're going to have to modify those inputs yourself. So why don't we move on to our next question? Here's a question, a more technical one. What tools can we use to compare VCFs from different calling methods?
So there are multiple tools out there. I think the best proven tool is called VCF eval from RTG tools. You can just Google that. It's open source. VCF eval. You give it a truth VCF, you give it a evaluation VCF, and then it outputs different statistics that you need. And it's quite sophisticated. It's not an easy problem, because the same haplotypes can be represented in different ways. And it actually creates graph representation of all that. So yeah, my recommendation is VCF eval from RTG tools.
Great. Are CNVs, copy number variants, are they analyzed in DRAGEN-GATK? I think the answer is no. We're only looking for SNPs and indels with this particular pipeline.
Yeah, that's right. So this particular pipeline is specific for SNPs and indels. We have DRAGEN has CNV analysis pipelines, and GATK has CNV analysis pipelines as well, both for Germline and for somatic use cases. And we have not at this time made any plans to combine those pipelines. So that'll be potential for the future. But at this time, we have separate pipelines. So there is not a DRAGEN-GATK one for CNV.
Yes, but if you are interested in running CNV analysis, if you go to the GATK website, we do have articles and tutorials on how you can do that for yourself using the GATK tools that we currently have available.
And there are similarly, like if you're interested in checking them out, there are Terra workspaces that also demonstrate how to use the CNV pipelines, etc. So if you search for GATK in the Terra workspaces list, public workspaces list, you will find that.
Thank you, Geraldine. So we have our next question here. It says, as the pipeline for DRAGEN aligner is being improved, will there be an update to the legacy GATK pipelines to use DRAGEN aligners instead of BWA? Well, I mean, first off, DRAGEN GATK is currently already in GATK. So once you update your package, it should already be included. But maybe Geraldine, you have some ideas of how the GATK package will be changed as a result of improvements in DRAGEN GATK in the future. What do you think?
Right. So I think you're referring to the fact that the DRAGEN GATK is functionally equivalent with a version of DRAGEN that's not the most recent version. And so there is some work underway to update the DRAGEN-GATK implementation to match, and I forget the exact version numbers. I don't, maybe James or Michael can jump in and give me the exact, but basically that's an ongoing work in collaboration with the DRAGEN team to port some of those more recent improvements into DRAGEN-GATK. We're also continuing to improve GATK itself separately and have a variety of improvements that come out to the tools. Typically, we have to distinguish kind of the improvements to individual tools in the GATK as a toolkit, and then the actual pipelines that are specifically shaped for a particular use case. DRAGEN-GATK is a particular pipeline that uses tools from the GATK toolkit. I hope that helps and did not add confusion, but that's basically how it works.
I think it was clear enough, Geraldine, but if you were confused, please go to gatk.broadinstitute.org and visit us on the forum. Okay, so next question we have here is about the performance of DRAGEN-GATK. Maybe we can put this to the other panelists, but how did we test the performance of DRAGEN-GATK on, how did we test it? Like, what were we using? And also, did we test it on FFPE samples?
So I guess my question, there's sort of, performance can mean two things, right? It can mean the actual scientific performance, or it can mean the runtime. I'm assuming you mean scientific. Well, first, assuming scientific, we have done the FE evaluation. There's a whole battery of different samples that we selected that were intended to sort of stress test it, I guess. There are samples that have, extensive PCR artifacts. There are samples that have been run PCR free. There's samples, we throw a bunch of, like, we specifically tried to select a wide range of use cases so that we would sort of drill down to potential error sources scientifically. Now, in terms of runtime, that's sort of a more sticky question, I guess. The performance, the runtime profiles of these two tools are pretty wildly different, right? Like, we built a WDL that runs in the cloud that is sort of cost optimized, and they have a version that's runtime optimized that can run in a very short period of time on one sample, but with a very expensive machine, right? So it's sort of not totally apples to apples to say, like, oh, how does our runtime compare to them, or how does it compare to the old GATK? I will say the open source DRAGMAP aligner is, as opposed to BWA, is not as optimized as BWA is. So we are paying a little bit in terms of runtime, and the haplotype color is sort of on par with where it was before. But that's like, this is sort of, we don't have extensive runtime numbers to sort of present at a public forum like this for these tools. Maybe that'll be something that's worth presenting in the future.
Great, thank you, James. And we have another question here, our last question, I think, which is, what's the availability of these tools outside of WDL? Is there a DRAGEN-GATK or similar, something similar to GATK that can be used in Bash or SnakeMake? Well, first of all, I mean, all of these GATK tools can be run on your terminal in Bash. So, I mean, you can do that. In fact, that's how I use it. And of course, as I showed you, we have availability of these workspaces in different platforms that you can use. I'm not sure about SnakeMake. I don't have experience with that. Can somebody jump in?
Well, I think the question is really, do we provide pipelines written in other languages aside from WDL? We don't. WDL is our kind of preferred and official language, and the reason is, like, that's the pipeline we use internally for the Broad's own whole genome analysis production facility, which is a very, like, high throughput use case. So we use WDL specifically, and so the GATK team develops in WDL. We do not provide pipelines implemented in other languages, but there are other groups that have done that. The thing is, it depends. So it is possible to find GATK pipelines I would say probably the most common that I've seen, Nextflow is the really popular one, I think, that has that's widely used. Various people I've written these pipelines. The question is like, how close are they to our best practices? So the DRAGEN-GATK one is the latest version of the best practices for whole genome SNPs and indels. Other people have pipelines that say they implement the best practices, and for many of these groups, so I think those are reasonable implementations. But you should, if you decide to use those, you should do some evaluation to decide if the results are high quality or not. So you can find a lot of pipelines out there. Some of them are implemented more or less faithfully compared to our recommendations. There are also people who implement our tools a little differently and have their reasons for doing that, sometimes adapting for their particular use case, and that's totally fair too. Just if you use pipelines that you find kind of out in the wild, do some evaluation to make sure that the results are going to be of good quality for your work. And that's what the pipelines are doing makes sense. That can be difficult. It's not an easy problem, but I would just say, like, the message, just don't just use it because it says it's the best practices. That means a lot. Just try to find an evaluation or do an evaluation to convince yourself that the pipeline is doing the right thing. But there are definitely groups out there that are doing good work, providing alternative versions in different languages, if that's something you really need.
And also, I believe that WDL was originally designed to be very approachable and easy to learn. So if that really is your hurdle, I think that it shouldn't be too difficult to just make modifications into the original WDL code.
Well, it's that's the intent of being the intent is to be accessible for people who don't have a strong programming background. Mm-hmm. I think as the optimizations have been added to make it like more economical to run, to make it highly scalable and things, some complexity has crept in, not the language itself, but some, like, the best practices, like the production implementation of our pipeline tends to be more complex than your basic example WDL. And it can be a lot for somebody who doesn't have programming background, to be fair. I would say, though, in my experience, people are more basing, well, there's part of it is your personal familiarity with the language is one important criteria to take into account. Like, if you want to be able to make tweaks to the language, is this something that's accessible enough and realistic, and it's not going to take you a whole bunch of time to learn a whole new language just to do that? The other question is, what is feasible on your local infrastructure? What tools are accessible? Some people don't have the opportunity to use Cromwell, and their sysadmins don't want to install it, and then they're kind of stuck. They have to use whatever is available, and so that might guide the choice as well. And we try as much as possible to support that and understand that we're trying to make it possible for everyone to run our tools in the best possible conditions, but we don't control what every institution makes available to their members. So we try as much as possible, but there will be a point where you might have to make some difficult choices or reach out for help. We'll try to help as much as we can. Sorry, Derek, go ahead.
No, that's perfectly fine. I just don't want to scare anybody off from actually getting their hands on the tools. So at the very least, try and do whatever it is that you want to do. And then if you have trouble, see if you can get help from us on the forums.
Yeah, I would say try it out. I was originally a microbiologist, no programming experience, and now I write WDLs, and I find it very accessible compared to some other stuff. Like Bash, I can't wrap my head around it. Maybe it's just me, but I find WDL a lot more accessible. But let's not get into a language, programming language.
No, that's fine. I don't want to get involved with that either. Okay, so another question here, just a clarification on the previous one. Is DRAGMAP and DRAGEN-GATK included in the newest GATK release?
Yeah. So DRAGMAP is in a separate code base that is owned by Illumina. So it's open source, but it is owned by Illumina, and they run that. They develop it. So and that's kind of how, if you can differentiate like the actual tool software and how it's packaged into containers, and then the pipelines, individual tasks in the pipelines will call containers or toolkits or whatever, right? And so you can have the aligner, just like BWA-MEM has always sat in a separate code base. The aligner is in a separate code base, but you can make a call to it. Did that make sense? I hope I didn't make it worse.
No, they're saying that it does make sense. Excellent. So before we wrap up, that's all the questions that we have right now. But before we wrap up, I've just entered into the chat a feedback form. And so we're going to be giving you some more resources before we leave. But I would like you all to at least open up that feedback form. It'll take you like 30 seconds if you care, and like 10 seconds if you don't. Yeah, that really, we do look at all the feedback that you have on our webinars. So I highly encourage you to tell us what you think and what worked, what didn't work. If you want to say, the host sucked, that's fine. If too many of you say that, then it'll make me sad, but we'll take that feedback into account. Okay, so I'll leave it to the panelists then to give any last words. Is there anything that you think that we haven't covered that maybe we should? I think that's a no then. Well, I mean,
you've covered a lot of ground, and I think that's a lot of information.
Okay.
But yeah, anybody who's interested in learning more about Terra, definitely look into the session, the interactive session we're having next week. It's two times 2-hour sessions, and that's very hands-on. Like, you walk through the exercises. We wait, like there's a lot more time given to hands-on, just so you can get used to the interface. So if you're interested in learning how to use it, so make sure to sign up. There's still some spaces available.
And how can people find more information about the future events that we have for Terra and sign up?
Yeah, so the Terra has an events page. Otherwise, you can also events page in the documentation that gets updated with upcoming events. You can follow the blog, terra.bio/blog. We post a lot about not just features and functionality, but also papers that people completed using the tools, sometimes new interesting developments in some of the technology and some of the tools, the capabilities, including GATK, but also other software toolkits that are hosted on Terra, which is bring your own, by the way. You can also bring your own tools, but there's a number of toolkits that are made available, kind of baked in, kind of like the GATK workspaces. So we blog a lot about those things. We also announce upcoming events, training events, conferences, and so on. And finally, we're very active on Twitter, so make sure to follow us on Twitter. GATK Dev is one, and Terra Bio App is the other one. We'll post that in the chat just now. And visit us on the forum as well.
Of course. And right now, I've also posted a link to a bunch of other resources that we have. First off, we've got the GATK website. Please visit there, as well as a link to our DRAGEN-GATK resources. That page will have all of our articles, presentations, and of course, videos that we've made related to that. And if you need more support using Terra, then that website is support.terra.bio. That's separate from the app.terra.bio website, but the support page has all of the articles. It has video tutorials that you can follow along with as well. So with that, I just want to thank you all for coming, and I wish you all good luck in using this DRAGEN-GATK workspace on your own. But of course, if you need more assistance, don't hesitate to contact us on the GATK forums. So with that, I'll let you go. Thank you so much and have a great rest of your day.